Thank you, Janet. As you can see, I'm presenting a paper with a large number of authors. In fact, you need 3 digits to enumerate all of them in decimal system. And this work is supposed to tell us a little bit about how well we can predict as a community protein function. So protein function, proteins do things, or maybe perhaps I should say that things happen to proteins. And regardless of your point of view, I'm going to use terminology that proteins carry out function. So for every experimentally annotated protein, perhaps there is a paper, or dozens of paper, or sometimes thousands of paper that describe what that protein does. And that description can go from multiple philosophical levels, from the biochemical level, what happens to the protein, to how that affects pathways, cells, tissues, and the entire organism. For example, for this particular protein, we can see that it's involved in protein binding, that it's phosphorylated at the biochemical level, then at the cellular level. It's a component of circadian clock mechanism. And finally, at the phenotypic level, a mutation in that protein causes advanced sleep phase syndrome. But we can't do it that way. We need to facilitate knowledge exchange, and we need to facilitate computing. Therefore, it's good to use ontologies. There's multiple ontologies for considering protein function, from functional catalog, enzyme, EC numbers. But the one that is most prevalent is by far the gene ontology, which consists of 3 directed acyclic graphs that describe protein function. And the two of those 3 graphs that are most important for this particular work is molecular function ontology and biological process ontology. Both of these ontologies have a large number of terms, 18,000. This is a couple of years ago, actually, in biological process, and about 8,000 to 9,000 in molecular function. This is one of the examples for a particular protein, hPNPase, that's involved in degradation of RNA. It has 7 domains. I will come back to that protein, but I just wanted to show you its molecular function graph. It's a directed acyclic graph that starts with molecular function, then propagates all the way down. When you see a dashed line, that means that there's multiple terms in between that are not even plotted. And just for the in terms of terminology, I'm going to call these terms at the bottom that do not have any descendants leaf nodes of the annotation graph. Now, there is an army of biologists that experimentally annotates proteins, and perhaps molecular biologists, and perhaps another small army of biocurators that makes sure that these proteins are well translated into standardized annotation. However, they cannot keep pace with the number of sequences that we currently have. Right now, we have 40 million sequences in Tremble. In SwissProt, there's about 500,000, and only some of those are experimentally characterized. But we are only at 7,000 published genomes right now. 20,000 are going on, and if it's really true that there's a million species right now, and possibly 100 million species, there is no way that we can keep up with annotating these protein sequences unless there's some breakthrough in experimental annotation. On top of that, all the experimental efforts are focused on a couple of model organisms, only 7 or 8. As you can see, maybe about 8,000 proteins in human are annotated in various ontologies. Yeast seems to be well annotated relative to its size. And other organisms, perhaps E. coli, there is a little glitch there because at some point they were removing sequences. But all these other organisms were annotated. There's only about 3,000 or 4,000 sequences annotated from all other organisms. So the whole annotation effort is focused on well annotating, understanding a small number of species, and then transferring this knowledge to all other species. And these proteins are annotated with certain biases. There's only some things you can do in one organism and other things that you can do in other organisms for ethical reasons or perhaps experimental convenience or the biases of funding agencies. On top of that, the situation gets difficult because some proteins carry out multiple functions. That is famous table from Jeffrey's paper in '99 about moonlighting proteins, where she listed a few dozen of those. We've later annotated more than that and shown that in terms of leaf nodes in molecular function that we can use as a proxy for multifunctional proteins, not quite well, but it's a rough estimate. About a third of the proteins have already multiple terms. Some of the proteins had about 10 terms or so, leaf terms. Oops. But in biological process, we can see that more than half of the proteins already have multiple terms, and some of the proteins, though with small probability, have around 50 terms. So it's very difficult to design a method that sometimes will predict 1 leaf term, sometimes 5, and sometimes 50. This really adds to the challenge of functional prediction. So the point of these previous slides is to persuade you that prediction of protein function is important and that the prediction of protein function is non-trivial. First, experiments cannot keep pace with the data. Understanding of protein function is important for understanding life at the molecular level, for understanding the mechanisms of disease. It also can provide clues for understanding evolution, and it has pharmaceutical importance. It can certainly prioritize experiments. But I also think there is some importance for the statistical inference in evaluation, because this is somewhat a unique problem in which an ontology, a graph, is assigned to a sequence. And of course, the problem is challenging because function is a multifaceted problem. There are different hierarchical or philosophical levels at which you can annotate protein. Experimental characterization is context dependent, depends on the particular experiment, depends on a particular organism. Proteins are multifunctional, they're promiscuous, bind multiple partners. Annotation is incomplete. There's no way that you can annotate a protein with everything that it does or it happens to a protein. It's simply very hard to enumerate all such experiments. And of course, there's noisy because sometimes there's curation errors or misinterpretation of experiments and so on. And finally, protein function is currently annotated, assigned to genes, because all the isoforms for a particular protein are assigned to a single gene. Therefore, once we start annotating different isoforms with different functions, it's probably going to get different. There's many methods that were designed. I'm just going to flash a couple of them. Phylogenetic profiles, gotcha that utilizes sequence and structure of the ontology, phylogenomic inference, inference from protein structure, and all these methods were designed on their sets of data. They were evaluated on a particular subsets of data, and it's really hard to say how well they do and how well we as a community do, and can we compare these methods. So we are trying to answer this question. How well are we doing as a community? And we thought the answer could be a critical assessment. By far, this is not a new thing. There's many critical assessments. There's even a review of critical assessments that just appeared this year. And even assessment of protein function is not new. It's been tried in CASP. There was MouseFunc. There was AFP, in which a couple of sequences were tried. But none of these efforts had enough sequences to provide really solid results regarding the ability to predict protein function. We also were interested in building a community in which folks would collaborate or perhaps compete just a little, but to, as a community, reach the goal of better predicting protein functions. Certainly learn from each other. So what is the task? The task is we provide a sequence or a bunch of sequences to the community. The community can use whatever data they like. We do not even tell them, nor do we provide any data for the community, and we ask them to predict a graph of a gene ontology graph. So subgraph of a graph. That is the task. It's very simple. So how did we do this experiment? The experiment was envisioned in fall of 2009. By March of 2010, we've assembled a very prestigious committee that guided us in everything. Stephen Brenner, Patsy Babbitt, Amos Bayrock, Christina Rango, Burkhard Ross, Sean Mooney, Michal Liniel. Maybe I should give credit to Ida Friedberg as well, who is one of those who envisioned the experiment. And Wyatt, Clark, Toby, and Tal are the persons who did the feasibility experiment and built the website. In this effort, we actually established that it will be possible to pull out such an experiment. We tracked the accumulation of experimental annotations in SwissProt and figured that we needed about 6 months or more to have some kind of reliable statistical comparisons. So we released about 50,000 sequences in September of 2010. So this is a 3-year project, basically, 2010, and we let people predict. We had a website, they could predict protein functions. Submission deadline was January 2011. This is the point that we call T0. I will say why. At that point, we collected SwissProt database, Goa database, and Go database. They're all overlapping, but contained slightly different protein annotations in them. We closed the prediction, we collected everything, and we let SwissProt accumulate new annotations. Okay? And once those were accumulated in December 2011, we collected those new annotations. We call it SwissProt of time t, and then we started the analysis phase. Basically, the set of targets that were used to evaluate prediction of protein function was SwissProt from December 2011, and then we eliminated all proteins that were in SwissProt Goa or Go in January of 2011. The target set was biased because curators have their own biases, funding agencies have their own biases. We had no control over the set of targets. But we thought we had a good representation of taxa in eukaryotic organisms. In total, we had 11 different organisms. But in terms of prokaryotic organisms, everything was based, coming from E. coli. Note that this is a logarithmic curve. So pretty much all sequences in eukaryotes are coming from human, and then the rest from mouse, and then from E. coli, and then other organisms contributed just a little bit. Still, we felt for eukaryotic organisms this was roughly a good representative set. And one way to show you that we did extensive evaluation is that roughly the distribution of leaf terms that I showed you earlier for molecular function is about here. 3/4 of proteins had a single leaf term. For biological process, roughly 50% of proteins had a single leaf term. By and large, we thought this was a reasonable set, but perhaps not statistically significant. We haven't tested it, actually. So now, what is the evaluation metric that we used? Consider this as a Go graph. Consider this to be a true function of a protein, perhaps DNA binding. We propagate this function to the root of the ontology. Now, let's say that the prediction is rRNA binding. We propagate that to the root of the ontology, and then we can derive two measures. One is precision, which is the fraction of predicted nodes that are correct, which is these two nodes, so 2 out of 5. And then we have recall, which is the fraction of true protein function that has been correctly predicted, so it's 2/3. We call this a protein-centric measure for evaluating protein function. There's also a term-centric measure that we perform that evaluation too. For example, you can say, okay, given that we are interested in DNA binding, can you rank proteins that are associated with that particular term? But this is a gene prioritization type of experiment. We were more interested in a protein-centric evaluation. Now, let's say that we have this precision and recall at 2/5 and 2/3. Obviously, predictors do not predict just a graph. They will provide you scores for each particular term. And in that case, you have to move the threshold and then provide predictions for each particular threshold. So that gives us a precision recall curve, 1 dot per each threshold that is tried. And obviously, this is the goal. If somebody can gain precision of 1 and recall of 1 when averaged over all proteins, we are done. We have perfect predictor. It is also important to, or perhaps it's interesting, to have a single statistic summary of predictors' performance. So what we've done is we took a harmonic mean of precision and recall at every point, which is called the F measure. And then for every threshold, we calculated the F measure, and the one where it was maximum, we call this F max. Okay? Now, for this evaluation metric, you can find a lot of complaints, but one appealing thing is that it's very interpretable, because we just measure the fraction of nodes correctly predicted or correctly retrieved, and then we find their harmonic mean. So it's easy to interpret. Now, participation. We had 23 PIs participated in the experiment, and because one PI could have multiple teams, as long as they do not overlap, we actually had 30 teams participating, and they tested 54 algorithms. There is no simpler way to summarize this number of groups. Most of them, they come from Europe. UK is the leader with five different groups. In the US, we had 2 groups from Asia, 1 from India and China. Okay. Now, before I start summarizing results, I would like to say that there's a lot of nuances in which should interpret these results. And no single evaluation metric that I show or ranking should really be interpreted as this is it. This is the clear winner and this is the clear loser. I will show you several of these results, and I think it's really important to get into the detail because otherwise you can get to incorrect conclusions. So this is the metric of Fmax for the teams averaged over all types of targets. This penalizes teams that predicted only on prokaryotes or that predicted only on eukaryotes. In case the group did not predict on, let's say, one whole set of organisms, we would give them recall of 0, and therefore they would be penalized and fall out of that curve. Still, it is informative to see what happens there, and as you can see for molecular function, the Jones group from UCL had fairly nice performance of 0.6. Argot is from Padova, Stefano Topo's group. Panzer from Lisa Holm group, Purdue group, Bologna group, Missouri group, Orengo group, Lichtarge, Brenner group, and Julian Goff's group. And then we can have similar rankings in a biological process. We compared these methods with two, we call them baseline methods. One is called naive. Every protein in that naive method is assigned the same prediction. And a particular term will have a particular score that was the prior probability of that score to be seen, of that term to be seen in the Swiss Prot database. On the other hand, BLAST was a simple application of BLAST in which we blasted the target protein against the database of experimentally annotated proteins. And now each of these proteins would have a sequence identity, local sequence identity, with the target protein. And therefore, Go terms that would be assigned to a protein will be assigned with scores that were equal to the sequence identity between target and the template protein. What we can see is that these methods significantly outperform BLAST, and we also can see that the performance in biological process is much worse. Now I can show you the same results as a precision recall curve. The blue curve is the Jones group. The red one is Argot. This is certainly more informative because as a biologist, maybe you're not interested in a simple average. Maybe you're interested at what happens at a recall of, let's say, 0.6. It means you will recover 60% of a true annotation of a protein. So what is the precision at which you can do it? It's about 60%. This is why Jones had 0.6. It's a harmonic mean. And so you can see every method, how it works for each particular threshold. For biological process, oops, maybe I can show you an example. This is the example from before. This protein has 7 domains. Each of them had a number of Go terms when looked over many proteins. And for example, this particular term, Jones group predicts it well at its optimal threshold. Orengo group predicts it well. This is the naive method. It predicts very shallow because it's based on priors. Here, Jones and Goff group predict well, and so on. For biological process, performance is not so good. You can see the ranking. I do not want to overinterpret this data, but the thick black curve is BLAST, and this one is naive. So sequence identity seems not to be a very good way to transfer biological process. Now you can stratify these over eukaryotes and prokaryotes. Suddenly the methods look different. In this particular case, we have Go-Struct method from Colorado group, Ben-Hur group, which they did not predict on prokaryotes. So suddenly they appeared as a very good performing group in eukaryotes. Similar happens with Lichtarge group for the prokaryotes. So again, I would just like to encourage you that look into details when analyzing these results. For biological process, very low performance for the algorithms for the eukaryotes. It seems like sequence identity cannot tell us much, and very few methods do just barely better than BLAST, while for prokaryotes, it was much better performance. And I'm not sure what to make out of it, but perhaps these types of organisms, because E. coli was most of the sequences, and it's a well-studied organism, it has a good interaction network, perhaps it had something to do with the results. Now, we reanalyzed these results with new data, because I showed you December 2011. Do the results hold up with the new data? Now, instead of 800 targets, we had triple the number of targets. The answer is quickly yes. There's about 1 percentage point difference in the performance of algorithms with April 2013. Similar happens with biological process. If you look at the results per organism, let's say I pick Arabidopsis. Again, we have a Colorado group. I can see this is a Confunk Sternberg group, and so on. If you look at E. coli. Here Jones goes on top, and so on. And of course, BLAST is over here, and this is naive. This is the really baseline. And for human, it's again a different set of results. So if you're interested in making predictions for a particular protein, I think it's a good idea to see where the protein is coming from and perhaps utilize the method that works best for those types of organisms. Now, before I conclude, I would just like to give a little plug that we are going to run CAFA experiment again. We're going to start this August with releasing sequences. We will collect them in January 2014 and then provide first evaluation in Boston at AFP-SIG. Again, we will test molecular function and biological process ontology, but we will add cellular component and human phenotype ontology. The second CAFA will enable us to test whether the improvement, if any, in prediction is due to algorithms or due to better data. And of course, we have good data sharing. We will not use your data without your permission. And you can opt out. If you don't have good performance, you can always opt out, except if you do really well. Then we need to publish your result and de-identify you. This is just a plug that we have already 2 accepted keynotes for Boston, Mark Gerstein and Fiona Brinkman. And now conclusions. What have we learned in this experiment? We think that we have a clear progress with, compared to the first generation methods, which are the straightforward uses of BLAST. All of the top algorithms significantly outperform BLAST in almost all categories. Therefore, an experimental scientist should be encouraged to use those tools instead of just using straightforward results from BLAST, even though the BLAST that we used is not quite what the biologists would do. They usually look at that data and look at publications and all of other things that maybe automated methods do by itself. Best methods usually integrate a variety of data. Over half of our methods have used more than one type of data, have used 2 or 3 gene expression data, protein-protein interaction data. In molecular function, sequence identity or similarity seem to be the best predictor of protein function, how to combine multiple hits. In the biological process, it seemed that protein-protein interactions and gene expression data played a very important role. We also think that machine learning methods seem to be helping. Now, in molecular function, we think that the performance is reasonably accurate. These models could be used by the community to prioritize experiments. But we were disappointed by the performance in biological process ontology. Much work is needed there. Therefore, there's many challenges to overcome. Databases are biased. We don't even know in what ways, certainly by experimental, not prejudice, but preferences by funding agencies and so on. Evaluation metrics are imperfect. I presented one that is interpretable, but it's certainly debatable how we should evaluate these predictions, especially since the full annotation is not available to us. We only have partial experimental annotation of a protein. One such metric will be presented by Wyatt Clark at 240. Function elucidation is difficult. It depends on the experiment. It depends on the organisms. And finally, for our tools to be available and used by the biologist, it's really important to have a tool that works and that is regularly updated. With that, I would also like to say that new ideas in how to infer function are also necessary, especially for biological process, where we could use better data, systems data in particular, and find algorithms that can make use of that data in principled ways. So finally, the acknowledgements: Ido Friedberg, Michal Liniel from the organizers, Amos and Sean in the assessment, Patsy, Steven, Christine, and Burkhard in the steering. Much of the work was performed actually on the right side. Wyatt, Gaurav, Tal, Alex, Toby, Burkhard Ross Group, a lot of advice and help in verifying the code and everything that goes into one such experiment, obviously all CAFA authors. And finally, funding agencies that allowed us to fund 11 students to travel to Vienna in 2011 and present their work. We hope to be able to bring some students to Boston too. Thank you. We tried some simple ways and it didn't work. They were not better than the best method. And the way was to actually perform some kind of cross-validation and use a statistical model that would combine predictions and learn from predictions. Didn't work